Hey, Dr. Free here. For this video, we will do an overview of the muscular system. The muscle organ consists of skeletal muscle fibers, and the muscle fibers are the muscle cells. It also consists of connective tissue layers, the endomysium, which is a connective tissue that wraps around the muscle fiber. The endomysium is areolar connective tissue. The epimysium and perimysium are also connective tissue associated with the muscle. The epimysium and perimysium are both dense, irregular connective tissue. And there are arteries, veins, and nerves associated with the muscle. The connective tissue, the three sheets of connective tissue, end up merging and forming the tendon, which is going to anchor the muscle to the bone and transfer the force, the tension that's generated in the muscle fibers to the bone for movement to occur. The arrangement are in bundles. So you have a muscle fiber and it has its endomysium around it. And the muscle fiber being a cell has a plasma membrane, a sarcolemma, around it. And then around the plasma membrane, sarcolemma, is a layer of connective tissue called endomysium. And then bundles of muscle fibers are grouped together in what are called fascicles. And around that fascicle, you have the perimysium. And then multiple fascicles are all bundled inside of the muscle. And around that is the epimysium. The arteries and veins are what make the skeletal muscle vascularized, and it's highly vascularized. There's a large number of arteries, veins, and capillaries. The capillaries are the smallest of the blood vessels. They are associated with the endomysium, and they're there for exchange. The muscle, being active in generating force and tension, is going to need carbon dioxide, it's going to need glucose, it's going to need to get rid of waste products like carbon dioxide, oxygen. So you need oxygen, get rid of carbon dioxide. And the capillaries associated with endomysium is going to be that site of exchange so that the muscle fibers, the cells, have access to the nutrients they need and can also get rid of the waste products. And then the nerve is important. The skeletal muscle is innervated. It's innervated by motor neurons. And motor neurons will send signals out to effector organs, in this case skeletal muscle, causing them to be stimulated, causing them to contract. Each neuron will have a long extension called an axon, and that axon will branch, and there'll be little knobs at the end called synaptic knobs. And the junction between the axon, the synaptic knobs of the axon, and the muscle fiber is called the neuromuscular junction. Looking at the microscopic anatomy, you take one of these muscle fibers and you enlarge it. So this is one muscle fiber, which again is a muscle cell. The muscle fiber itself is made up of bundles, and it's made up of bundles of myofibrils. That myofibril will extend the entire length of the muscle fiber, and the myofibril is made up of bundles of contractile proteins called myofilaments. The muscle fiber itself typically runs the length of the entire muscle, and it's a very long cell. It's formed by embryonic cells joining together. So in the embryo, you might have adjacent cells. They end up connecting. So you end up with this really long cell. And because each cell individually had its own nucleus, you end up with a really long cell that has multiple nuclei. So skeletal muscle is multinucleated. It has multiple nuclei for the entire muscle fiber, not just a single nucleus. The plasma membrane of the muscle fiber is called sarcolemma. So sarco is a term referring to muscle, so sarcolemma, plasma membrane. There are also another sarco, it's a sarcoplasmic reticulum. The SR, sarcoplasmic reticulum, It is like the smooth endoplasmic reticulum of other cells. It's just been modified. And it's modified, you can see it here, and that it forms a kind of a sleeve around the myofilament, around the myofibril, which contains the myofilaments. Make sure you keep those terms straight. And the sarcolemma also has some modifications. It has invagination. So if here's the plasma membrane or sarcolemma, it has these invaginations that come in, and then you have go back out to the surface. These invaginations are called T-tubules. See them there. The invaginations are associated with the sarcoplasmic reticulum, and next to the sarcoplasmic reticulum, you'll have endpoints of the sarcoplasmic reticulum that'll kind of come up next to the T-tubule, and these endpoints interact with the T-tubules, where the signal from the sarcolemma of the motor neuron is going to cause the sarcolemma to become excited. That signal is going to travel around the cell down these two tubules and interact with the sarcoplasmic reticulum. And at this point, the sarcoplasmic reticulum is called terminal cisternae. And the association of the T-tubule with two terminal cisternae is called a triad. And you can see it here. So you have the terminal cisternae, the membrane of the terminal cisternae on each side, and then you have the plasma membrane invagination, this T-tubule, and these membranes are in close proximity to one another. So what happens on the sarcolemma, the plasma membrane can come down this T-tubule and interact with terminal cisternae. And essentially what happens here is the terminal cisternae of the sarcoplasmic reticulum is where you're storing calcium, and when you have a stimulation of the sarcolemma, calcium gets released, and that calcium is going to be what causes contractions to occur. When you look at this a little bit closer, when you look at the sarcolemma, the plasma membrane, you can see that it has channels associated with it. There are voltage-gated sodium channels and voltage-gated potassium channels, and these will be involved in the excitation of the sarcolemma to be able to cause contraction of the skeletal muscle. When you look at the myofilaments, which are the contractile proteins, there are a couple different types. There is a thick filament, which is made up of myosin, and myosin is two proteins linked together, so it's kind of this tail and a head region, and then there's another tail and a head region associated, and then that's one myosin molecule. And then on the myosin, there is a site for ATP to bind to, and then there's also another site that actin binds to, and actin is one of the other types of myofilaments. And the actin is found on thin filament. The thin filament has two types of actin. There's a G-actin and an F-actin. The G-actin, the G, think globular, so the round balls are the G-actin, and then you end up linking them together, and they call that F-actin. Also associated with the thin filament is tropomyosin and troponin. The tropomyosin is kind of a thread-like, it's this green, it's a thread-like protein, and it aligns up with the G-actin in such a way that on the G-actin there are actually bonding sites on the G-actin for the myosin, and the tropomyosin lines up in such a way as to block those binding sites. And troponin is associated with the tropomyosin, and troponin is a globular shape structure protein, and troponin has binding sites on it for calcium. when the cell's at rest, when it's not been stimulated, you end up having the tropomyosin blocking the myosin binding site on the actin. When the cell is stimulated, then calcium is released. Calcium binds to troponin, causes a shift in the shape of troponin, which then pulls the tropomyosin off the G-actin, which exposes the myosin binding sites, and then the actin and the myosin can end up binding, and then that's what causes your muscle contractions to occur. The myofilaments, the contractile proteins that are located inside of the myofibril, the myofilaments inside the myofibril that makes up the muscle fiber, those contractile proteins are arranged in specific order, and they're repeating order. So the smallest unit is a sarcomere. And with the sarcomere, you have thin filaments and thick filaments in this specific arrangement that continues to repeat throughout the muscle. So you can see the repeating sarcomeres here. The ends of the sarcomere are these Z discs. They're kind of these little zigzag type lines will be the end of the sarcomere. Then the overlapping of your thin and your thick filaments end up resulting in bands or different regions where you can predict what type of filaments will or will not be present. And you have I band, A band, you have an H zone within the A band, and there's also an M line. So the I band will extend in both directions from the Z disc, and when you look at the I band, the only filaments that are located within it are the thin filaments. The thin filaments are inside of the I band, and it gives them a light appearance when you look at them under the microscope. So the A band is considered to be the central region of the sarcomere, and it'll contain the entire thick filament. So if you look at this A band region, you see that all of the thick filaments are contained within that A band. That A band has a dark appearance when you look at it through the microscope, and it's this alternating light, dark, that gives striations to the muscle when you look at skeletal muscle and also cardiac muscle under the microscope. This H zone is the center of the A band, and the H zone is the region where all you have is thick filament. So there's no thin filaments. This is the region just thick filament. The I band is the region of just thin filaments. The A band, you have overlap of thick and thin filaments on the edges of it. And the M line, this center piece here, that's going to be an attachment site for the thick filaments. When the muscles contract, you see these, the heads on the myosin stick up near the actin. So when they contract, those heads grab hold of an actin. So here'd be an actin molecule, here's your myosin. The heads grab hold, and then the heads swivel. And when they swivel, it slides the actin past the myosin. So when you have contraction, you'll shorten the sarcomere. So the sarcomere will shorten, but not all of these regions we talked about will actually change. With maximal contraction, the I bands can actually disappear as you have the myosin heads pulling the actin past, and you can shorten the sarcomere where those I bands end up disappearing because of that sliding past one another. The A bands will not change. The A band will stay the same size. It's the actin sliding past the myosin that causes the contraction. So A bands, which is the length of your myosin, isn't going to change. And with maximal contraction, the H zone can disappear. So the I band and the H zone can disappear with maximal contraction, and the A band is going to stay the same size. The skeletal muscle is highly innervated. There'll be numerous nerves, and it's neurons of nerves that run out to the skeletal muscle, and they are somatic motor neurons will run out to the skeletal muscle to control activity, be able to excite the skeletal muscle so you can have contractions occur. And the single motor neuron plus all of the skeletal muscle fibers that it'll end up innervating is going to be the motor unit. So with so there's the blue motor neuron comes out, and then that axon ends up splitting, and then you end up contacting. There'll be a junction that's not a physical contact. There's a little space there. The muscle fibers, and that site is your neuromuscular junction. Looking at the neuromuscular junction, so you have the axon of the somatic motor neuron coming in as part of your motor unit. This is going to branch, and then when it branches, there's this enlargement. The enlargement is the synaptic knob. So you can see this enlarged area here. And it comes in close proximity to the sarcolemma, to the plasma membrane of the skeletal muscle. But it doesn't actually touch it. There is a space there, and that's called the synaptic cleft. The portion of the sarcolemma of the muscle fiber that is in close proximity to the synaptic knob is the motor end plate. The synaptic knob houses vesicles, synaptic vesicles that contain acetylcholine. Acetylcholine is a neurotransmitter that stimulates muscle fibers. When an action potential, so when the neuron is excited, an electrical impulse called an action potential will travel down that neuron. It'll get to the synaptic knob, and when it gets here, it opens voltage-gated calcium channels, which means calcium then rushes into the synaptic knob of that motor neuron. And when calcium enters, it interacts with the synaptic vesicle, which causes it to merge with the membrane of the synaptic knob. And by exocytosis, acetylcholine is released. That acetylcholine will diffuse across the synaptic cleft, and then it interacts with acetylcholine receptors that are located on the sarcolemma of the motor end plate. So there's sections of motor end plate. All these gray structures are the acetylcholine receptors. The acetylcholine binds to it and will open the channels that allow sodium to rush into the cell. So sodium rushes in. And at the same time sodium rushes in, it also allows for potassium to leave the cell. And when you look at the motor end plate has a resting membrane potential. And that resting membrane potential means that if you look at the plasma membrane or sarcolemma, the inside of the cell will have a negative charge compared to the outside of the cell. When looking at the skeletal muscle, that resting membrane potential is minus 90 millivolts. Here we can look at the resting membrane. So the potential, that resting membrane potential is minus 90 millivolts, which means that that inside of the cell has a negative charge compared to the outside of the cell. And that's due to there being the sodium, potassium, ATP pump, which ends up pumping out 3 sodiums and bringing in 2 potassium. And then because it does that, it sets up a electrochemical gradient, and you have channels that allow sodium to passively diffuse in and potassium passively diffuse out. We spoke about this when we looked at plasma membranes in general with the cell chapter. So you may want to review how the resting membrane potential is created in relationship to sodium potassium ATP pump and the diffusion of sodium potassium. So that system is in place here. And also what's in place are these voltage gated channels. There's a voltage gated channel for potassium and also for sodium. And those voltage gated channels are on the sarcolemma of the muscle fiber. Remember, with the motor neuron, we had voltage gated calcium channels on the synaptic knobs of the motor neuron. And a voltage gated channel just means that a change, an electrical change, so change in that resting membrane potential will open or close these channels. Just like with the motor neuron, the action potential travels down this motor neuron, and it's a change. It's where instead of having the inside be negative and the outside positive, it's where you reverse that polarity. And so when that change in electrical current, that change in voltage arrives at this membrane, you can open these voltage gated calcium channels, allowing calcium to flow in. Same thing can happen here, and this can happen because the acetylcholine travels across the synaptic cleft, interacts with the acetylcholine receptors, and it causes there to be an influx of sodium and then efflux of potassium. As sodium enters into the cell, you get more and more positives, and you eventually will change the voltage enough that you can open these voltage gated channels. But when the cell is at rest, you end up having the minus 90 millivolts, and you have calcium stored within the sarcoplasmic reticulum. And with skeletal muscle contraction, going past this resting point, there's 3 components to it. You have neuromuscular junction, excitation of the skeletal muscle fiber. So you have to excite the skeletal muscle fiber, and then you have an excitation contraction coupling, and this will deal with the sarcolemma, T-tubules, and the sarcoplasmic reticulum. Then you have activity within the sarcomere where you have cross-bridge cycling. So looking at what happens at the neuromuscular junction, you have the action potential arrive, you open the voltage-gated calcium channels, so calcium rushes into the synaptic knob. The synaptic vessels are going to then be activated. They're going to release by excitosis acetylcholine. Acetylcholine is going to diffuse across the synaptic cleft. It binds to acetylcholine receptors and causes sodium to move in. It also causes a little bit of potassium to move out. It's primarily the sodium moving in. You'll get enough change here that you activate an action potential. And that action potential is propagated. It can travel, and it'll travel down the sarcolemma, and because it's traveling down the sarcolemma, it travels down the T-tubules and comes in contact with the terminal cisternae of the sarcoplasmic reticulum. And here, the change, the action potential, that change in voltage, will cause calcium to be released. So you open voltage-gated calcium channels. Calcium is released from sarcoplasmic reticulum, and it floods the sarcomere. So it goes into the sarcomere. When it gets into the sarcomere, that calcium is going to bind to troponin, which is going to cause the tropomyosin to move and expose the myosin binding site on the actin, and then you get the myosin and the actin to form a cross bridge in that swiveling motion, resulting in shortening of the sarcomere and your contraction. And then this just focusing on that neuromuscular junction excitation, what we just spoke about, the calcium will enter through voltage gated calcium channels. You get the release of the acetylcholine. That acetylcholine then binds to the acetylcholine receptors on the motor end plate. We're looking at the excitation contraction coupling. You have the development, it's called an end plate potential, EPP, at the motor end plate. And this is due to the acetylcholine binding and getting the sodium and potassium to end up diffusing. Sodium rapidly diffusing into the cell and potassium diffusing out. That EPP is a change in the resting membrane potential, and you go from about 90 millivolts to about minus 90 to about minus 65 millivolts. So the inside is becoming less negative or more positive by going from minus 90 to minus 65. And that change is your end plate potential. That change, getting to minus 65, is what will trigger the voltage gated sodium and potassium channels to open. And first you get voltage gated sodium channels opening, so you get even more sodium moving into the cell, so the inside of the cell becomes less and less negative, more and more positive, until you actually have the inside of the cell having a positive charge. And then you also have the voltage gated potassium, voltage gated potassium channels opening, and the potassium will move out. And when the potassium moves out of the cell, you actually end up taking the cell back to minus 90. So what's happening here is you're going from the inside of the cell being negative with the outside being positive to changing so that the outside is negative, the inside is positive, and then this is primarily due to the sodium movement, and then you go back to that normal state where the outside's positive and the inside's negative, and that's primarily the potassium movement. This sodium, this is called a depolarization, making that inside less negative, more positive, and eventually becoming positive. And then when you restore the original state, that's a hyperpolarization. And that alternating of depolarization and hyperpolarization is also an action potential. So that action potential is propagated. It travels across the sarcolemma, down the T tubules. When it gets to the T tubules, it's going to cause the release of calcium from the terminal cisternae. As this action potential travels down the T tubules, it interacts with voltage-sensitive calcium channels on the sarcolemma, and those will then interact with the calcium-releasing channels, which then cause the calcium to be released from the terminal cisternae of the sarcoplasmic reticulum, allowing it to flood the sarcomere. So looking at the action potential, it's important to know what's on this graph. So there's time, and then there is the membrane potential measured in the millivolts. So at rest, when there's nothing going on, no activity within the cell besides normal maintenance, you are at minus 90 millivolts. Something will happen to change that, and this change is also called a depolarization. And with the skeletal muscle, that depolarization is due to acetylcholine, and that depolarization is actually your end plate potential. Same thing. That end plate potential causes the change from minus 90 to minus 65. And when it does that, that minus 65 is a threshold. And everything past the threshold, this whole event, this rise, depolarization, repolarization actually goes a little bit past. That whole event is your action potential. And that action potential, it's all or none. So if you reach threshold, you're going to get an action potential. If you don't reach threshold, you won't get an action potential. And once it starts, it'll continue. You're not going to stop it. And the other thing about the action potential, besides being all or none, it's also propagated. It can move along the sarcolemma, along any membrane that has a resting membrane potential. So this rising state, this depolarization, that's going to occur because you have voltage-gated sodium channels on the sarcolemma. They open, sodium rapidly enters. You go from minus 65 to plus 30. Plus 30, the inside of the cell is now positive. At rest, the outside was positive, the inside was negative. When you have the action potential, then you reverse that. The outside becomes negative, the inside becomes positive. It's actually this change, is positive, negative, negative, positive, that allows it to be propagated. Because so this is at rest, and this is where the action potential is. Right next to the action potential, it's at rest again. So you have this opposite charges, positive, negative, negative, positive, and you just get flow of ions, which then causes this area, this adjacent area, to be stimulated, and you get an action potential here, and it just keeps continuing all the way down the sarcolemma. The other part of the action potential is this repolarization, and the repolarization is due to the voltage-gated sodium channels closing and the opening of the voltage-gated potassium channels. And the potassium then moves back into the cell, and you go back to your resting state. So you reverse from the positive 30, you go to minus 90 millivolts, and you bring the cell back to the resting membrane potential. The calcium will cause cross-bridge formation, and that cross-bridge formation is between the myosin head and the globular actin. So what happens here is the calcium binds to the troponin. So calcium comes in, troponin are the little blue molecules, calcium binds to it. When the calcium binds to it, it's then going to expose. See these actin sites, the myosin binding site on the actin is now exposed. When you expose them, you get cross-bridge formation. So the actin head binds to the myosin molecule. And the actin head, the myosin head is already activated. It has ADP and inorganic phosphate attached to it. Notice there's no ATP. Like ATP has already been used, which means in this point, the myosin has potential energy. Like it's cocked. It's ready to do something. So when it binds, you get the myosin head swiveling. So you can see there's this change. The myosin head is kind of swiveled here, and it swivels towards the center of the sarcomere, and it pulls the thin filament as it does so. In this motion, it's called a power stroke. And in the process of doing this, swiveling this power stroke, the ADP and the inorganic phosphate are going to be released. So at this point here, the myosin and the actin are bound together. So the myosin head swivels, the ADP and the inorganic phosphate are released, but the myosin head actually is not released. The myosin head is still attached to the actin. In order to get the release of the myosin head, you have to have ATP. So ATP will bind to the ATP binding site on the myosin head, and that'll actually cause the release of the myosin head from the actin. And then when that myosin head is released, you want to reset it or recock it, and that ATP bound here and caused it to be released, the ATP is then going to split into ADP and inorganic phosphate. And when it does that, that energy that was in ATP is used to recock or reset that myosin head so that it is all ready, it's all prepared to be able to bind again and form cross bridge. And this just keeps cycling. As long as there's calcium present, this will keep cycling unless the muscle reaches fatigue. But in general, if calcium's present, then you continue to have this cross bridge formation occurring. So the calcium has the actin site exposed, so myosin can bind to it. You end up getting the power stroke. You release the ADP in inorganic phosphate. ATP then comes and binds to release the myosin head. ATP is split to reset the myosin head, and then it can bind again and just keep cycling. So this is a summary of what we spoke about, talking about the neuromuscular junction, the excitation-contraction coupling, and then what is happening with cross-bridge formation. So you should be able to take this and draw this out, label all of it without referencing anything, and that is a great way to see if you know the information. And if you can do that without referencing anything, then you know that you're prepared to take quizzes and tests. If you can't do that, if you have to keep referring back, then you know that you need more assistance and more time learning the information before you take quizzes and tests. So looking at the cross-bridge formation, we know that we have to have ATP involved in the process of cross-bridge formation and shortening the sarcomere and contraction. So when you look at the skeletal muscle, there's some ATP available inside of the muscle. It doesn't require oxygen. It's a very limited amount of ATP, only a couple of seconds worth of ATP. Once what's available is used up, then the process of glycolysis, again, doesn't require oxygen. You have a rapid production of ATP, but it's a less amount of ATP than what's formed with the aerobic respiration. Fuel's going to be typically glucose. That'll last for about a minute, and then in order to get enough ATP to continue contractions, then you have to have the aerobic cellular respiration. Oxygen being required, it's going to occur within the mitochondria. You have a slower production of ATP, but significantly greater amounts of ATP. And then fuel, the pyruvate from glycolysis. And then you also have fatty acids and amino acids can be used as fuel in the aerobic cellular respiration. It's just a nice visual. So available from the ATP and phosphate transfer, it's about 5 to 6 seconds, maybe 50 meters worth of a run. Glycolysis, you get a little bit longer. It's a short-term energy source, maybe 50, 60 seconds, like a 400 meter. And then if you're doing anything beyond that point, then you need the aerobic cellular respiration, say like the 1500 meter run. There are different types of muscle fibers, and the different types are based on the type of contraction and the primary means of ATP supply, where it's getting its ATP from. With the type of contraction, just looking at whether it is a fast or slow, primary means of ATP, looking at where it's coming from. Is it primarily from glycolysis, or is it primarily from an aerobic cellular respiration? So looking at those combinations, the type of contraction and primary means of ATP, you can have different types of skeletal muscle fibers. A slow oxidative, type 1, a fast oxidative, type 2A, a fast glycolytic, which is a type 2B, and then they have these corresponding traits associated with them. Well, the next step with the muscles is now we understand how the physiology of how the contractions occur. What is the outcome of those contractions? And that outcome is muscle tension. And tension is the force generated by the muscle. And here we have a myogram, and myogram is just a graphic representation of changes in muscle tension when the muscle's stimulated. So you have a muscle, you stimulate it, and you get this graph. And you can stimulate changes in voltage and changes in frequency. So if you measure this, a single stimulation, you will get this brief contraction and relaxation period, and that is a muscle twitch. So single brief contraction and relaxation due to a single stimulation is your twitch. There is the point at which the stimulus is applied, and then there is a latent period. there's a lag time from the stimulus being applied to actually seeing an increase in tension. And then you have the contraction period where you see this increase in tension, and relaxing period where the tension starts to drop and decrease. That contraction period is going to be the repetitive power strokes, and the relaxation period that'll start with the release of the cross bridges, and that's going to be due to calcium being put back into the sarcoplasmic reticulum. It's important to notice that a twitch is not an action potential. So again, twitch is not an action potential. The twitch is a measurement of tension of the muscle fiber. So looking at tension within the muscle fiber. The action potential is a measurement of change in millivolts. So it's a current change, and it's looking at the sarcolemma. When you look at these two graphs, initially they look similar. You've got this little flat point, you got a rise, a drop, return back. And same thing, you have a flat point, a rise, a drop, but then it drops down farther, and then it returns back. So this, they're visually looking at them, they do not look the same. And then they also do not represent the same thing. So you want to be really careful that when you're looking at graphs, that you are sure whether it is a twitch or whether it's an action potential. With the muscle tension, you can look at changes in stimulus intensity as well as stimulus frequency. And here we're looking at intensity. So if you have a continuous increase in the strength, the size of the stimulus, if you increase the intensity, then you see an increase in tension. And then you also will see an increase in motor unit involvement, and there'll be increase in numbers, so you have more motor units involved, and then you have motor units that have more muscle fibers, so larger motor units being involved. So that means that we refer to the increase in tension due to increase in intensity as motor unit summation, because we're looking at increasing the number of motor units and the size of the motor units that are involved. On this graph, you can see that as you move along, that you're having an increase in the voltage, so increasing the intensity, going from like 2.5 to 4 or 5, that sort of thing. So as you increase the intensity, you can see that there's a corresponding increase in the muscle tension. So low intensity, low muscle tension, higher intensity, higher muscle tension. And then that correlates back to the number of motor units. So if you have just small motor unit involvement, small fibers, then you have a lower level of tension. As you increase the number of motor units and you add larger motor fibers, larger activity of the muscle, then you will see an increase in tension. So the stimulus frequency can also be looked at. And if you have a frequency that it's rather infrequent, then you get a twitch and you see that your tension stays the same. And the frequency is such that it's applied, so the tension, you get the increase in tension, you get the relaxation, you go back to normal, and then you apply the frequency stimulus again. So you're applying the frequency at such a slow rate that you're not seeing any kind of change in the size, the tension associated with the twitch. But if you increase the frequency, then you can see some changes. So as you go on here, you're increasing, you're applying faster, 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 faster. Initially, the first change you see is this wave summation. So instead of the twitch occurring and going back down, the twitch occurs, and then you get a stimulus before it gets to relax, and you get a higher tension, and then another stimulus before it relaxes and higher, and you just keep increasing. If you increase the frequency enough, then you see the tension gets closer and closer, and you get eventually where it fuses, and then that's the tetany. If you continue then you would get fatigue occurring. The contractions in the muscles can be categorized based on the force generated by the muscle and the resistance that must be overcome, or the load that must be overcome. And there are two types. There's isometric contraction and isotonic contraction. Isotonic contraction is when the muscle tension is greater than the resistance, so the muscle will actually shorten and movement occurs. So picking a child up, that would be where you have a movement occurring. The isometric is where the skeletal muscle tension is less than the resistance, and you're not going to have movement occur. And you can remember it, the isometric, think like metric, measurement, iso, it's the same. There's no change in the measurement. So there's no change in the muscle length. Another example of isometric would be pushing on a wall. You're not going to have movement. You're not going to have a change, isometric, due to the difference between tension and resistance. With the muscular system, we have focused on skeletal muscle, but of course there's also cardiac muscle and smooth muscle. Cardiac muscle with 169, there'll be a lot of discussion on cardiac muscle, and also smooth muscle comes up a good bit with 169. This is a good time to familiarize yourself with the comparisons between skeletal, cardiac, and smooth muscle to start building that foundation, so that when you do get to looking at cardiovascular system and then looking at various systems with association for smooth muscle, that you have an idea of the differences and associations, where they're found, appearances, sources of calcium, that sort of thing. So you want to be familiar with this as a general comparison between the three different types of muscle. That is the overview of the muscular system. If you have any questions, let me know. And again, this is just an overview. You want to use the module activities in SLO list to get the depth of information that you need.